planetary model for oxygen, and it has like H2O, H2CO, CO2, and all that stuff. But I don't know how to
Gotcha. All right, 1 second, and I'll show you here. Let's see. See if I can do this. Obviously, I'm gonna have to rush through this because we don't have much time. But if this explanation doesn't do it because it was so quick, then obviously you can still visit me during um, office hours.
All right.
All right, let me see here. I'll try to pull up a whiteboard. I don't even need to share it because you guys hopefully will see it on my screen. Oh, there we go. Can you see my whiteboard, like a big white box on the top left?
Yes.
Okay. So let's use nitrogen, for example, instead of oxygen, because I can't give away the answer. If you were to draw a planetary model of oxygen, nitrogen, you're just drawing, basically the most important thing is showing me that you understand where the electrons are and how many there are. So, I might put, let's see, nitrogen, 3. It's early yet. I'm trying to remember how many, the atomic number of nitrogen. So 8 is oxygen, so 7. Okay. So nitrogen's got 7 protons. So I maybe draw 7p there. Maybe even draw 7n to say, yep, there's 7 neutrons. So there's the nucleus. Then I'm going to draw the electrons. So we've got the first ring, and the first ring holds 2 electrons. So I'll just draw 2 electrons. All right. Well, if it has 7 protons, I know it has 7 electrons. So I know I still need more electrons drawn. So I keep drawing this. The second ring. All right. So I already have 1, 2. So 3, 4, 5, 6, 7. And then that is the planetary model of nitrogen because, again, it has 7 protons. So I know it has 7 electrons. So I drew 1, 2, 3, 4, 5, 6, 7 electrons. And I followed all the rules. 2 on the inside and, no more than 8 on the outside. So now I'm done drawing nitrogen. Now, if the next question, and this is a little bit different than the question on the lab, but it's enough to push you in the right direction. The next question, if it were to say, all right, draw me a nitrogen with a -1 charge, then I need to think, okay, well, I've already got my nitrogen. So I need to draw this with a -1 charge. And what does a -1 charge mean? Well, that means it has an extra electron. So I'll just draw an extra electron, and then boom, I'm done. Did you get that?
So it says -1. You just add like one more on the outside of it.
Exactly. However many negatives it says, that's just how many more electrons you're going to put.
Okay.
And for the sake of teaching, even though you don't need this for that particular assignment, hold on, let me undo this. All right, we are back to, this is the original nitrogen. Now, if it were to say, all right, draw me a nitrogen with a +1 charge, and it didn't, I'm just doing this for the sake of teaching, you would take away one of those electrons, right? Because there's a positive charge, that means you got one less negative thing. So you would take one of those electrons away. So yes, that is how you would do a planetary model.
So it's like kind of backwards.
Exactly. Well, yeah, depending on how you look at it, yeah. Because when the thing that makes it backwards is when you say +1, then you're taking one away, and when it's -1, you're adding one. But just remember, electrons are negative, so that's why it's backwards, so to speak. Okay. Now, I might have time for me to briefly show you how to draw the things. I'll give you the first one. Actually, let me think of something different. I wish I had a little bit more time. Basically, you're just going to draw the molecules, and because they're all carbon-based, well, almost all of them, you're just going to want to put, shoot, let me think. Let me look something up really quickly.
Did you get started early?
No, I'm talking about something else. Good question. I'm just helping her out before the class starts, and I'm recording it. That way people can refer back to it if they want. All right, C6H13NO2. C6H13NO2. Where did my whiteboard go? There we go. I don't have enough time. So yeah, Reed, I guess if you want help or anybody else, visit me during office hours and I can give you some hints on how to draw those molecules. If you look them up, you'll see stuff like this, but that's not what I'm looking for because no one's taught you how to draw those. So yeah, just see me during office hours if anybody needs help with that.
All right. Thank you.
You're welcome. All right, now back to this. Good morning to everybody. And yeah, I'm glad Vance asked. No, we didn't start early. I was just helping somebody. So where are we at here? I don't need that. Okay. Yeah, we've got a couple minutes left. Not enough time for me to finish what I was trying to start, but also not enough time or too early to start. We will do attendance the way we usually do. So if you're online, listen for the keywords. If you're watching this because you weren't able to watch it live online, then obviously you'll have a set of questions to go with this. And just to clear it up, I don't know. I don't pre-plan the pre-words or the attendance words, just so you guys know. So I don't know how many attendance words there's going to be. I felt bad. I didn't unintentionally last time. There was 3, and I could tell some people were worried that they missed a word because it's usually not 3. So I don't know. I could start planning ahead, I guess, to make sure that I have the same amount, but I'll worry about that moving forward. Any questions about anything? Because our time's about to start and we are recording, so I don't feel bad answering questions. Yeah, it's 8. All right. Well, before I get started, I'll remind you guys, I know it's tough, especially with me not physically there for lab. So when you guys need help, make sure you come to me during office hours. I posted the link again last night on that announcement. I'm always here. You don't have to do any of this alone, especially lab. So if you're having problems, like I don't understand how to do this, or even if you're just lost, like I don't even understand what's due, come to me. Come visit me during office hours. We'll figure it out. But anyway, that being said, let's jump back into the lecture. Can someone please confirm that you can hear me and see the PowerPoints?
Yes.
Perfect. Thank you. So yeah, we're already done talking about carbs. Now we're getting into lipids. And so far, the only thing we've told you about lipids is that they are hydrophobic, meaning they don't mix with water. And at that point, that's when I finally told you that carbohydrates are hydrophilic, meaning they do mix with water. You can dissolve them in water. So anyway, now that we know the lipids are hydrophobic, and we also know they're not like these other 3, because these other 3s are polymers. And remember, a polymer, it's a long chain, right? It's a long chain of something made up of repeating monomers, right? So we knew all those carbohydrates, they were all just a bunch of glucose for the most part, they were a bunch of glucose molecules put together. Lipids, they are larger molecules with smaller parts, but they're not necessarily where it's like the repeating parts, like the same small parts. And you'll see what I mean, but that's not going to be important for the lab. It's just something to keep in mind. So anyway, the first thing you need to know about lipids, other than what I've already told you, is that lipids themselves are broken down into 2 categories. So remember, carbs were broken down into 3 categories. We had monosaccharides, disaccharides, and polysaccharides. Lipids, there's 2 different types that we will talk about. The first one is fats. The typical fat, also called a triglyceride. And you should know that word. You should know that basically fat or triglyceride in this chapter, actually in the semester, because it's the only time we're really going to talk about it, those 2 words are interchangeable. Now, of course, if you're talking about the fat on your belly, I won't even get into it because that is related. But so yeah, just remember that a fat is a triglyceride. Why is it called a triglyceride? Well, does anybody know what this prefix tri means? Any guesses?
3.
Yeah, exactly. 3. It has 3 fatty acids connect to something called a glycerol molecule, and that'll make sense when I show you. But yeah, for the exam, you'll need to know that. Know that it's 3 fatty acid molecules attached to a glycerol molecule. And there's a question like that on the study guide. There might be a question like that on the exam because you do need to know the parts of these things we're going to talk about. And these fatty acids, these are going to be important parts a little bit later, and you'll see why. So anyway, moving on, a typical fat is a typical fatty acid is very long, and because of that, it's very energy rich. Because basically, the best way for me to explain it is, the more energy that goes into building a molecule, the more energy there is to be released when you break it. So obviously, the bigger a molecule is, generally speaking, the more energy it takes to build it. So that's something to keep in mind. How much energy? Well, your book tells us it is twice the energy of a carbohydrate. Now, that's a pretty good, important number, but this isn't a dietary class. This isn't a health class, so I'm not going to ask you to memorize that. But how about this? This will be the first word for attendance. Twice. Twice is the first word for attendance. Anyway, another thing about typical fats, as you might guess, they are stored, or as you might know, they are stored in adipose cells. And you might be wondering what adipose cells, and that's just the proper term for a fat cell. So there you go. And again, you won't need to know that either. For the exam on this slide, I would say the most important information is that the word fat in this context is interchangeable with the word triglyceride, and that it is made up of a glycerol molecule and 3 fatty acids. Any questions? All right, moving forward. If you download the PowerPoint, you can watch this happen. So here's a glycerol molecule, and here's 3 different fatty acid molecules, and they're all going to come together to make one molecule. And it's a little early, so I'm not going to ask you guys to guess, but what you should know, if I was going to ask you, even though I've just shown you this, and say, hey, what kind of reaction is that? And you should know, because it's bringing things together, that is a dehydration reaction. Because again, for the exam, you will get a question like that. I'll probably show you some reaction you've never seen in your life. But it doesn't matter, because you'll know if I'm bringing smaller things together to make a larger thing, then it is a dehydration reaction. And here's a picture of it instead of an animation. And again, the other hint is that it's producing a water molecule. So anyway, yeah, what you end up with is this one glycerol molecule and these 3 fatty acids attached. And again, you can see how that they're very long, right? And remember, octane was just a chain of, 1, 2, 3, 4, 5, 6. So if you were to cut this off right there, that's an octane, right? That's the exact molecule that you would find in your gasoline. That's what makes it go. So imagine you've got chains even longer, and 3 of them. So there's a lot of energy here. All right, some essential functions. You might need to know this for the exam, and you might already know it. Think about fat. Why does your body have fat? Not like what led to it. Like, what good does it do? Why do you even need fat? Why does your body need fat? Any guesses? Okay. Insulation, that's a good one. And usually people guess that last, so good guess. Yeah. Usually when you think of insulation, you think of, or at least I do, I think of animals that live out in the cold, like polar bears and things like that, or whales that live in really cold water. So yeah, they need insulation. But yeah, humans do too, especially when you think about the fact that humans haven't always been clothed and living in, houses. So yeah, cushioning's one of them. What's another one? Or here's it. Let me lead you to it. And I was going to not ask you this, but it's a good hint. What makes you fat? How is it that you get fatter? What very simple thing do you do that makes you fatter?
Eating.
Huh?
Eating.
Yeah, eat. Specifically, when you eat more calories than you burn, right? So what does that mean? What possibly could be another function of fat? Hopefully that's a good hint.
Stores energy.
Yes, stores energy, right? So really, that's when you see somebody, if you look at my belly, you can be like, wow, he's got a lot of stored energy. That's a nice battery. And then, so we said, okay, there's another one, and this picture is the hint. So we said stored energy, we said insulation, and what else? What's going on with the fat on this foot? No guesses? How about the fat on your rear end?
Cushion.
There you go. That's it. So energy storage, cushioning, and insulation. So you might need to know that for the exam. I'm not quite sure. Hopefully it's easy to remember, because just think about the fat on your body. And in my opinion, the hardest one, again, to remember would be insulation. But there you go. So any questions about that? All right. Here's where it gets a little bit more complicated. And these are words you've heard before. And this is pretty important because it's very important for your health. So let's clear things up. If a fatty acid, so remember, we're talking about those long chains that come off the glycerol molecule. If a fatty acid has fewer than the maximum number of hydrogens, it is called unsaturated. And if it has the maximum number, it is called saturated. And the way you can remember that, I think, is, think about like somebody who just came in from a long run and they're covered, like just completely covered in sweat. You might say to that person, wow, you are saturated. And what in that context, what that means is they have so much sweat, on their shirt, maybe, that they just the shirt can't absorb any more sweat, right? They're saturated. That's all the sweat that shirt can absorb. And the same would be said for this. If it's saturated, that means it just has all the hydrogen molecules, the hydrogen atoms it can attach, right? There is no other place to attach a hydrogen atom here. Now here, for the unsaturated, you could say, yeah, if you were to break these double bonds, so it's only a single bond, then this one could attach to a hydrogen, and this one could attach to a hydrogen, right? So that is what the difference is, or that's the first difference between saturated and unsaturated. And there will definitely be I shouldn't say definitely. I plan on there being a test question that comes from the study guide that says a saturated fat is saturated with what? And the answer is hydrogen. So that's the important thing so far to remember. When I say it's saturated, I'm saying it's saturated with hydrogen. Now, that's talking about the fatty acid. Now, if we're talking about health and diet, and you hear someone say, this meal has 3 grams of unsaturated fat, or this meal has 5 grams of saturated fat. Now, when you talk about it like that, instead of saying fatty acids and you're saying fat instead, then there's different rules. So for you to say, like if you were to look at a nutrition label and it says, 5 grams of saturated fat, what that means is, I'm going to try to draw it. There's the glycerol molecule and the 3 fatty acids. That's so horrible, but there it is. That means that all 3, 1, 2, 3, all 3 are like this. They're saturated. That's the only way. If even one of them looks like this, if even one of them is unsaturated, of course I kind of drew it that way anyway, then that whole thing is then called unsaturated, right? So it can only be saturated if all 3 are fat, or all 3 are saturated. Does that make sense? Okay. Then we have something called polyunsaturated, which is, and you can see this unsaturated, it's got a little kink here, right? It bends because of that double bond. Well, polyunsaturated would be like this, where it bends because of a double bond, but then it bends again because of another double bond, and then maybe it even bends again. And the same thing could happen here, right? Bend up, maybe bend again. The point is, maybe bend that way. The point is, when they're unsaturated, the more unsaturated they are, first of all, the better. And we'll talk about why. But they get this really weird straight shape, right? If it's saturated, it's always going to be nice and straight. So are there any questions so far? All right. Most animal fats are relatively high in saturated fat. In case you didn't know, it is the saturated fats that are bad for you, which is why, generally speaking, you want to limit your intake of animal fats in your diet. The reason they're so bad, because they easily stack, therefore they are solid at room temperature. So again, if I'm trying to draw an unsaturated fatty acid, it kind of looks like an E the way I draw it, that would be easy to stack because it's nice and straight. I can bring in another one and like put it right there, Oh, I'm so bad at drawing, but try to imagine that was straight instead of all wonky, right? See, those things would stack. And because they can stack up, that's what makes them solid at room temperature. So if you remember at the beginning of the chapter, there was that little picture of the kid eating chocolate. And I said, notice that it's a solid bar of chocolate, but on his hands and its face, it's all melted. And that's where this comes from, right? Saturated fats, they can be solid at room temperature, like butter, for example, but put a little bit of heat to them and they melt. But again, the fact that they are so easy to stack at the molecular level, that's what makes them solid at room temperature. And that's also what makes them so bad for you, because they clog your arteries. So for the exam, for the exam, you might want to know that it's saturated in fats that are bad for you. And the reason that they're bad for you is because they're easy to stack, right? They're easier to solidify, and that's going to clog your arteries. I also used to associate that with animal fats. Any questions so far? All right. Most plants and fish fats are high in unsaturated fat. So now we're talking about the opposite. So generally speaking, at least for the exam, and if you're going to associate things, generally speaking, you would think of animals as saturated fat and plants and fish as unsaturated fat. Unsaturated fats are liquid at room temperature, and they're actually really good for you. And there's also something, I don't even think your book talks about it. There's something called PUFAs, polyunsaturated fatty acids, and those are really good for you. Things like walnuts, you get those in walnuts, avocados. So anyway, if you're into it, look into it for independent work. You could look into the benefits of a diet high in polyunsaturated fatty acids. It's really good for you. It's good for your liver. It's good for your heart. Anyway, any questions so far? All right, here's a slide, or I think this is from your book. It's definitely from your textbook publishers. It shows the different fat, the different types of fat. Here we've got corn oil versus palm oil versus butter. You can see that the one that has the most saturated fat is butter, which makes sense. That's an animal product, right? It comes from milk. Palm oil, it's only 50% saturated fat. And you have corn oil, which is only 13% saturated. Now, this is nothing to memorize. If anything, and I probably won't do this, but if anything, I might give you this picture on the exam and say something like, which one has the most polyunsaturated fats? And you would look and be like, oh, yeah, corn oil has the most polyunsaturated because it has 50. That one has 9. That one has 3, right? You might have to decipher something like this, but that's it. Any questions about that? All right. Pretty simple stuff, right? All right. So there is a way to turn an unsaturated fat to a saturated fat. You've probably heard these terms before. Now, if you don't already, you will know what they mean. When you're doing that, when you're taking that unsaturated fat, that one that doesn't have all the hydrogens it can, and you're converting it to a saturated fat, so you can adding more hydrogens to it, that is called hydrogenation. And you might need to know that for the exam. Probably not, but maybe. Hydrogenation. It's in the name, right? You're adding a hydrogen to it. As a matter of fact, let's go ahead and make that the other attendance word. Hydrogenation. Anyway, so you might be wondering why even do that? Like, why would food companies even go through that? Why would they spend the money and the time and the effort of making something saturated? Well, it helps foods not spoil quickly. And there's a good independent word topic. How is that? What is it about being saturated as opposed to unsaturated that helps it last longer? It's good for frying. Another good independent word topic for you. What makes it good for frying? It can make solid at room temperature. We already talked about why that is. So there's no reason to look that up, right? It's easily stackable on the molecular level because they're nice and straight. And I guess, the benefit of that is think about something like margarine. If you're making something that's supposed to be like butter and you're making it from corn oil, then, you can't really cut into oil, right? So you want something solid. And that's why, that's another reason why you might go through hydrogenation. And the bad thing is, though, it can create something called trans fats, which are very bad for your health. And I'm not going to ask you any questions about trans fats because because if I remember correctly, that's all your book says about them, is that they're very bad for your health. So I'm not going to take the time to talk too much more about them. You can look them up for independent work if you want. What are trans fats? Why are they so bad for your health? Where can you get them? Things like that. Any questions about this slide? All right. Again, this is a good time to remind you, since we're talking about going from unsaturated to saturated, because the question that you will see is, when we say something is saturated with fat or saturated fat, what are we saying it is saturated with? And the answer is hydrogen, right? It's saturated with hydrogen. All right, another type of fat. This is a good one. So when I was talking about PUFAs, the polyunsaturated fatty acid that I don't think your book talks about, this is one of those, right? An omega-3 fatty acid falls into that category. They're very healthy. They reduce heart disease. They reduce arthritis symptoms. They reduce irritable bowel syndrome. They're good for your liver. The list goes on and on. They're really good for you. And they're found in oily fish and nuts. But of course, they're still fats, so they're still high in energy. So, if you're on a diet, and when I say diet, I don't even like using that word. If you're trying to lose weight, you still want to burn more calories than you're taking in. So you have to be careful when you're eating these. They're still good for you, but you still, even good stuff, if you're taking in more calories than you're burning, it's going to be stored as fat, and that is unhealthy for you. All right. Any questions about omega-3 fatty acids? Okay. And again, you can look any of this stuff up for independent work if you want to dig deeper into it. Found in fish and nuts. All right. Well, which fish, which nuts, which nuts have the most, which nuts have the least, which fish have the most, which fish have the least, so on and so forth. Here's just a little picture from your book of everything we've talked about. Again, generally speaking, you should associate saturated fats with animal products and being unhealthy. And generally speaking, you should associate unsaturated fats with plant and fish, and you should consider it healthy, generally speaking. And of course, there are exceptions, because here we go. Trans fat, right? That is unhealthy. Margarine, if I remember correctly, we now know it's unhealthy. Anyway, any questions about this? All right, you guys are nice and easy. So again, we were talking about fats, and fats falls under the category of lipids. And I told you when we started, there's two different types of lipids that we're going to talk about. The first one was fats. We're done talking about that. Now we're going to talk about the other type, which is steroids. So all the things I just told you about fats, you need to know about them, but you also need to know they fall under the category of lipids. So you might get a question, and I'm going to say, what is a fat? And so the choices might be carbs, lipids, proteins, and nucleic acids. And the answer is lipids. Same thing with steroids, right? Because lipids fall under steroids fall under the categories of lipids. And they are also still hydrophobic, just so you know. All right. Steroids, they're different from fats. Remember, we're still talking about lipids, but they're not fats. It's a different category because they have a completely different structure and function. Specifically, their structure, and you sort of need to know this. I mean, you should be able to identify it. You don't necessarily need to memorize the words. All right, 4 carbon skeletons fused together. But if you see something that looks like this, you should recognize it as a steroid. But yeah, this is the basic shape of all steroids. It's 4 of these carbon skeletons, these carbon rings fused together, and they're always fused together in this fashion.
Hey,
What makes
so that picture that you had showed us when you was talking to Kyandra?
The picture I showed you?
Bro, it's Keandra. It's not that hard one.
Which
he was talking to Keandra. All right, so there was a picture that you showed him, but just like that. You said we don't know that yet.
Huh. Yeah, so this is a steroid. Yeah. Exactly. And this is the basic shape. They all have this basic shape. The way they're different, the way one steroid is going to be different from the other is the functional group. And those are the things that stick off of it. And I'm going to give you some examples of that here in the next few slides. So any questions yet? All right. So here's a steroid that you will probably need to know for the exam. Obviously, there's so many different steroids that are out there, but you only need to know the basic ones. And the most basic, important one is cholesterol. The reason it's so important, well, the first reason is because it's a key component of cell membrane. So the things that make up the outside of your cell, you wouldn't have them without cholesterol. So without cholesterol, you wouldn't have your cells. Therefore, you wouldn't be alive, right? So obviously, when biology, the scientific study of life, cholesterol is important because you wouldn't have life as we know it without it. For the context of this chapter, the more important bullet point is this one. Cholesterol is the base steroid. So that every steroid you have is basically comes from cholesterol. Your body takes cholesterol and it makes some changes to it. And here's a good example. Here's what cholesterol looks like right here, this top one. And you can see with a few adjustments, you can make 2 different steroids. So hold on, let me skip ahead. No, it's not on there. Okay. Can anyone guess what this steroid is on the left versus on the right? Which one this is? And there's one hint in the pictures. I don't expect you guys to look at the steroid like, yeah, I know that one. I can tell by the way it looks. But the hint is in the pictures.
Male and female?
Yep, you got a male on the left and a female on the right. So any guesses on which steroid this might be on the left?
The male is testosterone and the female is estrogen.
Perfect. Yep. Not that you need to know that, but yes. So yeah, here we have testosterone on the left, estrogen on the right. And, those are our sex hormones, and they're similar but different. They have similar but different functions. But obviously, and also when you look at them, they have similar but different shapes too, forms, right? So you can see here, this one has a double bonded oxygen, where this one has a single bonded oxygen that's attached to a hydrogen. Here, again, this one has a double bonded oxygen. This one has instead an oxygen bonded to a hydrogen. And that's it. That's the only difference between those steroids. And just those slight differences make the difference, well, and some other things, make the difference between a male and a female. So for the exam, what you need to know is that cholesterol is the base steroid. And then you need to know that testosterone is associated with males. Estrogen is associated with females. Now, that's for what you need to know for the exam. For your own knowledge, females have both estrogen and testosterone. They just generally have more estrogen, and the amount varies from one female to another. Same thing with testosterone. Males have testosterone and estrogen. They just generally have more testosterone. And again, the amount varies from one male to the other. Also, the amount varies in the same male from one day to the other, from 1 year to the other. So anyway, any questions about cholesterol or testosterone or estrogen? All right. I think when most people hear the word steroids and they're not in a biology class, they're usually thinking about this, which is synthetic anabolic steroids. And I'm not going to ask you anything about this. Excuse me, I'm still sick. But I'm going to go through it very quickly because your book did. And because, again, most people, when they think of the word steroids, this is probably what they're thinking of. Synthetic anabolic steroids. These are variants of testosterone that mimic some of the effects of the testosterones. There are some good uses for them, which is why they exist in the first place. They treat diseases, basically any disease that would waste away your muscles. Not any disease, but some diseases, if it were to waste away your muscles, or waste away at even other cells, your synthetic anabolic steroids would help combat that. Even things like poison ivy. I don't know if you guys have ever gotten poison ivy really, really badly, and they gave you a steroid pack. And that's why. But anyway, so those are good uses. Obviously, they can be abused. Anything that has good uses can be abused. And some people do that to build up muscle quickly. But that's not necessarily a good thing because it can cause serious physical and mental problems, such as violent mood swings, depression, liver damage, high cholesterol. Hey, that makes sense, right? Because cholesterol is the base steroid. Shrunken testicles, reduced sex drive, infertility, so on and so forth. And again, this is not a health and human performance. This is not a health class. So I'm not going to ask you any of these questions. But are there any questions about this slide? Okay, good. That means we are done talking about proteins. Lipids. which brings us to the next category, which is called proteins. And you're probably all very familiar with proteins, and we're going to break these down into 2 main bullet points. Now, I want to be clear with something. When we talked about carbohydrates, there were 3 bullet points. There was one for each different type of carbohydrate. When we talked about lipids, there was 2 different bullet points because we were talking about 2 different types of lipids. For proteins, we're not really going to do that. I mean, there are different types of proteins. They're very, very varied, versatile molecule, but we're not going to break them down into the different types. I will discuss it once, but we're not going to do that. So these bullet points are not broken down into the different types of proteins. The way this is going to work is first, we're going to really talk about the monomer. So for the carbs, for example, I just told you, hey, the monomer is a glucose, C6H12O6. I showed you a couple pictures. That was the end of the story. And you didn't need to know much about it other than the fact that you needed to know that glucose was the monomer. The proteins is a little bit different. We're going to talk a little bit. We're going to go in a little deeper and talk a little bit more about the amino acids, the monomers of the proteins. And then after that, we're going to talk about the protein shape. So there we go. Proteins. Proteins are polymers of amino acid monomers. Know that bullet point, because as I told you before we even really got started in this chapter, when we were talking about monomers and polymers, I told you that was going to be the theme of the chapter. That for each one of these things, you would need to know the monomer and you need to know the polymer. So right now I'm telling you what the polymer is, or the monomer. I'll tell you what the polymer is later. But right now you need to know that the amino acid, that is the monomer of protein. So what are proteins made of? They're made of amino acids. The same way starch is made up of glucose, proteins are made up of amino acids. Now, these are really important. How important? Well, they make up over 50% of the dry weight of most cells. So remember, the number you do need to know is that your cells are 70 to 95% water. So if you were to get rid of all that, then your cells they're a majority of protein. So the fact that proteins make up so much of your cells, that should tell you something, right? Obviously, they must be important. The reason being is they are needed in almost everything cells do. And we'll talk a little bit about that in this chapter. And then later in the semester, that's going to be a very important concept. And your book says they're the most sophisticated molecule in your body. For the exam, the most important thing here on the slide is this top bullet point right here. The proteins are polymers that are made up of amino acid monomers. Any questions so far? Okay. So here's what I told you. Remember I said we're really not going to get into the different types of protein the way we talked about the different types of lipids and the different types of carbs, but I am going to discuss it. So really quickly, we'll talk about the different types, but then we'll just leave it at that and not come back to this. So you have structural proteins that provide support. So things like hair, eyebrows, fingernails, hooves, horns, right? Those are all structural proteins. Then you have things like storage proteins that provide amino acids for growth. So obviously eggs need a lot of protein because if you're going to build more if you're going to build more proteins, then you're going to need those building blocks, which are the amino acids, right? So eggs are high in that. Same with some seeds, right? Because those little seeds are going to grow up to be a new plant. So you're going to need a bunch of stuff for growing. Contractile proteins. I think a lot of times when people think of protein, this is what they think of. So your muscles, right? The things that help you move stuff, that's a contractile protein. Or right here, when you eat meat, right? That is a different animal's contractile protein. Then you have things called transport proteins. Talk a little bit about those in the next chapter. Those are basically things that help get in and out of your cells. And we'll talk about that later, like I said. And then there's the really important one as far as the exam is, as far as the semester is concerned, and they're called enzymes. For now, you don't need to know much about them, but later we'll really start talking about enzymes. But basically, enzymes are these proteins that help chemical reactions happen. So anyway, yeah, without any of these things, we wouldn't have life. So any questions about this slide? All right. Here we go. Let's talk about it. Let's talk about the monomers. Let's talk about these amino acids. So for all proteins on life, on Earth, right? Every living thing that has, well, every living thing has protein. So for every living thing, all this protein, there's a common set of 20 amino acids. So if you were to look at the proteins in your body, like break them down to their monomers, completely break down all your proteins, you would only find 20 amino acids. If you were to do the same thing to an elephant or to a bacteria or to a sloth or, I don't know, a palm tree, you take all the amino acids and break, all the proteins and break them down to their monomers, which are amino acids, you would only find 20 amino acids, and they would all be the same. So that's another one of those unity of life things, right? All living things share these same 20 amino acids. So obviously, if there's 20 amino acids, each one of them is going to be different. And they are, but obviously there's also some similarities. And this right here shows the similarities. And you do need to know this. Like, I'm not going to ask you to draw it, but you need to know the parts. So every single one of these 20 amino acids has this: the central carbon. Remember, carbon bonds 4 times. So if you can remember that, then you can remember that, all right, there's 4 things that I need to know here. So the carbon is always bonded to a hydrogen. It's always bonded to something called an amino group, which should be easy to remember because it's an amino acid. It's also bonded to something called a carboxyl group. And it's also bonded to something called a side chain. So every single one of the 20 amino acids, this is what it's bonded to. And it's always bonded in this order. So it's not like there'll be an amino group here and then a carboxyl group where the H is, and then the H where the carboxyl group is, right? But that doesn't, actually, I shouldn't have even told you. It doesn't matter because I'm not going to ask you to draw this. If anything, I might ask you the 4 parts of an amino acid. And again, it's the amino group, the hydrogen, the carboxyl group, and the side chain. So then the question is, all right, if all 20 amino acids have this, if they all look like this, then what makes one amino acid different from the other, right? How do we have 20 different amino acids? And that's what I'm about to explain to you. But are there any questions about this slide? Okay, and again, I just want to make sure you're aware. You do need to know that there's an amino group and a hydrogen, a carboxyl group, and a side chain. But you don't need to know that a carboxyl group is this carbon double bonded to an oxygen, single bonded to an oxygen, blah, blah, blah. You don't need to know this. You don't need to know what a carboxyl group is or what an amino group is. Just know that that's one of, those are the parts of an amino acid. All right, so here's everything I just said, but it's in writing. But now I'm going to differentiate a little bit here. So yes, we still have the carbon is still single bonded to these things: a carboxyl group, an amino group, and a hydrogen atom. But I also said that it is, let me back up, bonded to something called a side chain. And that's what this part is right here. The side chain is the part that makes it different. Because no matter which amino acid you're looking at, the carboxyl group's always going to be the same. The amino group's always going to be the same. The hydrogen atom's always going to be the same. So the only way that these things are different, the thing that defines the amino acid, so to speak, is the side chain right here. This is the thing that makes an amino acid different. Sort of like, this is really sort of, I'm kind of stretching it here. The same way I said an element is defined by its number of protons, right? That's how you define it. That's what makes it different. That's what makes a hydrogen different than a helium, for example. Well, and this is important, you need to know this for the exam, the thing that defines an amino acid is its side chain, which makes sense because the amino group is always the same. The hydrogen is always the same. The carboxyl group is always the same. So the only thing that makes one amino acid different than the other is what its side chain is. So any questions so far? All right. So each unique side chain gives that particular amino acid its special chemical properties. Because remember, we said that earlier, it's the side chains. It's very often the functional groups, and this is a functional group too. that's what kind of makes it do whatever it does chemically. And you'll see what I mean when I show here in the next slide. But yeah, so again, it's the side chain that defines the amino acid. It makes one amino acid different from the other. And it also gives it its unique properties, right? Is it hydrophobic? Is it hydrophilic? Is it polar? Is it non-polar? Is it charged? Is it not charged? All these different things. So here we go. Let's take a look. Here's an example. Now, these are real examples. There really are amino acids called leucine and serine. You definitely don't need to memorize it. I'm not going to ask you. I'm not going to say which one had the triangular side chain, leucine or serine. You don't need to know that. This is just an example, but it's a good example. Because one thing, and I said this before, I don't remember when, it wasn't too long ago, but I wanted to let you guys know that with chemistry, it isn't, I mean, the way I taught it, the way we teach it in 101 is it's like all about the electrons, right? The electrons are, you're trying to get a full outer ring. So it's all about the electrons when it comes to chemistry. And that's true. But another thing that your book doesn't really talk about, and I want to talk about, is sometimes it very often is like puzzle pieces. Sometimes, like when you're trying to squish molecules together, they either fit or they don't. And so their shape does matter. So sort of going back to the saturated fat idea, right? The reason saturated fats are solid at room temperature is because they're nice and long and straight. And you can easily just stick those things together. So the same can be said for amino acids. So when they have different shapes, they just fit things differently. So that's one thing to keep in mind that your book doesn't talk about. But then let's talk about some of the things that your book does talk about. This thing is hydrophobic, right? Leucine is hydrophobic. So if you were to mix this with water, that thing would twist, if there was a whole line of amino acids, which is what a protein is, then this particular part of the protein would try to stay away from the water because it is hydrophobic. Meanwhile, this part is hydrophilic, so it would be attracted to the water. Or even better, forget about those water. Keep in mind that hydrophilic things and hydrophobic things, they are attracted to each other. Like a hydrophobic thing is attracted to another hydrophobic thing. A hydrophilic thing is attracted to another hydrophilic thing, but they repel from each other. So if this were on a protein, these two things would try to stay apart, right? Because they would repel each other. And this will be important later because a protein, it's all about what shape it's in. So you have to think about all these different things that are going to attract and repel the amino acids from each other. Anyway, so those are just some examples. Again, this one's hydrophobic. This one's hydrophilic. Some of them might have a charge, like a positive charge, which means they would be, repelled by other positive charges. Some of them might have a negative charge, so on and so forth. There's all kinds of different things that make them unique. But anyway, that's just one book example. Any questions? All right. So what happens is these amino acids are brought together like a long chain of amino acids. And when we do that, we call those peptide bonds. And that's a word you might need to know for the exam. So when we bring amino acids together, those are called peptide bonds. And I don't remember if your book tells you, but these peptide bonds, they're just covalent bonds. I'm going to abbreviate because it's really hard for me to write like that. So yeah, peptide bonds are just covalent bonds, except, it's biology, so we have to keep things complicated. So when we're talking about specifically covalent bonds between amino acids, we call those peptide bonds. And honestly, I wouldn't even teach that to you if it wasn't in your book and if it didn't help you remember this one word that you definitely need to know, which is this: polypeptide. So you already know that the monomer of a protein is an amino acid. I already taught you that. I already said you needed to know that, but I never told you what the polymer is. So the polymer of a protein is called a polypeptide. So again, that's one of the big reasons they even taught you this word peptide bond. Because if you can remember many peptide bonds, hopefully that'll help you remember that it's protein. So any questions about that so far? All right, here's where it gets a little bit more complicated. And I'm not necessarily going to test you on this, but this part right here. A functional protein is one or more chains or one or more polypeptides that have been twisted and folded and coiled into a molecule of a unique shape. In other words, a polypeptide is just a chain of amino acids, right? Just a chain of amino acids. It's not considered a protein until it gets folded up and shaped into whatever particular shape it needs. See, that's not like starch and cellulose and glycogen, right? It didn't matter what that was, just a long chain of glucoses. It doesn't matter if they were twisted or curled or whatever. It just was what it was. Starch is starch no matter what. Proteins are not proteins until they assume their final shape. So here, can anybody tell what this is a picture of? Right here. Any guesses?
It looked like a basket. Well, I don't know why I said that so loud, but I don't know what that is.
Anybody else? I can see it does look like a basket. Of course, you could use the same analogy with a basket that I'm about to use too, but no, it's some sort of like knitted blanket. Can you guys see it now that I've told you what it is? Or maybe a scarf laying over a chair. Can you guys see that now?
Yeah.
Okay, like a scarf or a blanket made of yarn.
An Afghan or something.
There you go. Yeah. So you can think of these things in the same way. If you have a ball of yarn, or even if you unball it, if you just have a string of yarn, what do you have? You have yarn, right? That's it. Now, if you can do this and knit it into something, you might have yourself a set of mittens or a scarf or an Afghan, whatever. It's still a piece of yarn, but once you twist it and fold it into that shape that it's supposed to be in, it is then considered a blanket or a scarf or whatever it is. Does that make sense? So it's not a scarf until you folded it and twisted it to be that shape. And even when it is in that shape, it is still technically yarn, but you now consider it, a scarf or whatever. Is everybody clear with that?
Yes.
Okay. Same with a paper airplane, right? If you were to take a piece of paper, it's just a piece of paper. But then when you fold it in the exact right way, it is then considered a paper airplane once it is folded into that shape. Now, granted, that paper airplane, it is still a piece of paper, but we now consider it a paper airplane because it has been folded into its proper shape. And that is the relationship between the word polypeptide and protein, right? A protein is a polypeptide, but it's a polypeptide that has been folded and shaped, and it is in its proper shape, right? It's all about the shape for the protein. Anyway, here's a picture of two amino acids coming together. And hey, who'd have thunk it? You're bringing things together. That's called a dehydration reaction, right? Bringing together this monomer and this monomer to making a larger, a polymer. And look, you're producing water. And there you go, dehydration reaction. Again, this is just an example. So when you're studying, please don't memorize this picture. But yeah, that is just another example of a dehydration reaction. So any questions about this? All right. So here's an interesting thought. There are thousands of proteins in the human body, but there's only 20 amino acids. So then the question is, how is that possible, right? How do we have, and if you want, by the way, this is a good topic for independent work. If you feel like writing like 2 sentences or 3 sentences, something really short, just look up that. I was wondering how many amino acids there are, how many proteins there are in the human body. And according to this source, there are this many proteins. Anyway, so then the question is, like, how do we have thousands of amino acids, but we only have thousands of different proteins, but only 20 different amino acids? And your book uses this good analogy of the English alphabet. Think about how many words there are in the English language. Well, we only have 26 letters. So how do we have only, millions of words in the English language when we only have 26 letters? And it's all about how you're putting those letters together, right? It's the same thing with proteins. Sure, there's only 20 amino acids, but it's all about how you're putting those 20 amino acids together. And you can put them together in so many different ways. So that's how we have these thousands of different proteins. Back when I made this PowerPoint, which is when I first made it, I make changes to it, but back when I first made it, I think there was 1,025,109 words in the English language, but you definitely don't need to know that for the exam. It's just an example. If you download the PowerPoint, you can watch this little clip right here that talks about the primary protein structure. The next word for attendance will be determines. Right there, so you can see it. That is the next word. Anyway, the amino acid sequence is what determines the three-dimensional structure. In other words, going back to what I was saying earlier, a protein isn't a protein until those polypeptides are folded and until they're the shape that they're supposed to be into, they're not considered a protein. But once they're in that shape, they are considered a protein. And this is what it's all about, right? It's all about the shape and what determines its shape. It's not like they just randomly fold up together or ball up together. There's something that does it. There's something that dictates the way these things come together. And it is the amino acid sequence. And I've already hinted at this because I said, let's look at this, for example. This thing, I don't even need to tell you the chemistry on it, but this thing has an S, and then this one has an S. What you don't know, and what I'm telling you, is that these S's are attracted to each other. So this may have been one long straight line of amino acids, but this one's attracted to this one. So now we have this curve in the amino acid, right? And then this isn't the case, but let's say this one has a positive charge and this one has a positive charge. So now these things are repelled from each other. So now the thing curves this way, right? So it's all these different intricate interactions in between the amino acids that give it the shape. And remember, it's the shape that matters. Without the shape, it's just a long strain of amino acids. So proteins have jobs to do, like all these enzymes and the structural proteins and the proteins that move your muscles, right? Or that are your muscles. So it's all about the shape. But if they don't have the shape, they don't do their job. And what makes their shape? It's all these little side chains and how they interact. It's all about the amino acid sequence. So any questions about this slide? All right. So here on the left, we have a picture of a polypeptide. It's only curved because they had to fit it on the screen. Other than that, it might as well be straight. And what happens is, this is the way on the right. This is what it really looks like once it curves up and folds up the way it's supposed to. It's two different ways of looking at it. There's nothing to memorize here. The point is, these amino acids, they have these unique side chains that repel them from some and attract them to others. And then they fold up and you have different shapes. And that's 8:50. So that is the end of this lecture. I will be back on for office hours at 9 o'clock. So please, if you need me, come see me. I know some of you need help, so let me help you. Any questions while I'm still recording? All right. Well, in that case, I will log out of here. And like I said, I will be back at the other link, the last link that I shared on Google Classroom. I'll be back on office hours for that at 9 a.m. And I'll try to stay past 9 a.m. because I'm here. Everybody's here at my house because of COVID. So if you come and I'm not there on the screen,